So the next plenary talk will be given by Professor Igor Meglinski. He is the head of the Biophotonics and Biomedical Imaging at the University of Otago in New Zealand. And he's one of the plenary speakers for the SPIE part. So please, Igor, you have your 30 minutes.
Hello. Sorry. Okay, so good morning, everyone. First of all, I would like to say thanks to organizers. First of all, to organizing such nice conference as we saw yesterday, especially. So, and I in particular want to say thanks to Christina Kurachu, who invited personally me for this conference. So, and it's very nice to be here, so, and very great place, so, and I'm very pleased to deliver this talk. So, first of all, for these few days being here, I'm actually found that name Igor is quite popular in Brazil. But I'm not sure if you guys know that name of Igor is very, very scientific name. Oops, wait a second. Okay. Okay, so if you look at this cartoon, you will find that Igor is basically associated with Dr. Frankenstein and using the electromagnetic radiation for creation of life. So I'm also involved in the, that sort of research, as I believe is most of the Igors. And we also use this electromagnetic radiation as a light for diagnostic purpose. And of course, in terms of this conference, you know that diagnostic is important, and very early diagnostic of cancer is especially the way, you know, to save some lives. And our group actually is okay. So our group is based in the University of Otago. It's the most southern university in the world. So, and it's a Dunedin city. So, and for us, it's quite convenient because I represent the physics department, but we're working with the School of Medicine, and School of Medicine in the Otago is the biggest school in the region. So, and thus we're involved in a number of activities, and on this slide is just summarized what we are doing. But again, one of the important part of our research is develop new diagnostic modalities, and particular for imaging and early diagnostic of cancer. And that's what I will talk today about. But again, so here's we also worked with many other technologies. So and basically, I'm a physicist, okay? So I'm not a medical doctor, and when I started to understand the problem, you know, what is actually diagnostic is and how it works. So we found that the new word for us is a biopsy. And when I'm just discussed with the clinical doctors what is a biopsy, with the practitioners, and they explained me that basically what happened when the patient, after some, you know, some time in life, and seen on the surface of skin something suspicious, and he or she basically not aware of what is that. And the patient is coming to GP, general practitioner, and the general practitioner is also, in most cases, also not sure what is that. And what they do, they simply will cut that suspicious place with one of the possible way, and then they will send these pieces of tissues to the lab, and they will put it in a special cassette, the one of the standard way of diagnostic, and they will prepare the tissue blocks, paraffin-embedded tissue blocks, and then these blocks will be collected from many patients, and then they will send it to special lab that basically uses outsourcing from the hospitals, and using the microtome machine, they will prepare a very, very thin slices of tissues with about 3-5 microns, and they will put it on the standard microscopy slides. And you see, so that's quite sophisticated, you know, job to do, because, you know, tissues could be, we can simply lost some fragments of the tissues. And then the hospitals need quite high training personnel to prepare the samples. And also what is important, so they will produce a number of slides. And these slides takes, we need to take time to analyze the images on that slides. And sometimes it's not easy to make a judgment and understand, so is it cancer, non-cancer, pre-cancer, or what is that? And as a result, if we have a number of patients, so we have a queue, cost, delays in treatment, and so on. So, and one of this challenge, again, coming to us, and we thought what we can suggest to overcome that challenge. But on the other hand, when I spoke with these clinicians and pathologists, I asked them, okay, guys, could you tell me what do you see really when you look in your microscopies? And that's an example of images that basically they see through the microscopy. And I asked them, okay, how do you know which one is cancer? And they said, the one pathologist said, okay, so I have a experience like about 40 years. We said, okay, I have no experience at all. Okay, give me the secret. Tell me, what is that? How you do know that? And they explained me, okay, so that when they see some difference in size of cells and some, quite woggy boundaries and so on. So what I want to say, it's quite subjective, actually. So what they see and how they do the judgment, is it cancer or non-cancer? But on the other hand, as for physicists, I understood that if that people, trained people, able to see with a simple magnification the tissues and able to judge cancer or non-cancer, that means that we potentially able to suggest a solution to, for example, to using polarized light or circularly polarized light that could be automatically, give me an answer, is it cancer or non-cancer? And now I will just explain to you why actually do I think that the cancer can work and where the progress at the moment. Okay, basically just to those of you who are not from the physics side, imagine if we have this light just like sunlight or any other, so it's a non-polarized light. But when we put the polarizer, it's produce the light that propagates along the one plane, and we call that line is the polarized light. So, and thus, when we produce the one polarized light, so when we use another polarizer that we call analyzer, so we will be able to see the black and white pattern, so that shows is polarized light propagates through or not. So, and why actually polarized light is good for us? So first of all, when we're talking about the polarized light, so we basically think in terms of wave. And when we're talking about the polarized light, we're able to mathematically prescribe clearly through the sine wave, which is oriented in X or Y, for example, here. Or if we consider two different components, so we're able to represent it in a more general case. Means we're able to, with that simple mathematics, we're able to prescribe any state of polarization. Okay, so, and basically, when we think about what we see on the detector, we basically measure one or another component with core or cross-polarized light. And this is also have a very special sense because in that case, we're able to describe these components in terms of so-called Jones vector, and that represents all possible polarization states mathematically. Okay, so, and we're able to see how these components are changed, and we're able to produce the special equation that describes this. So, circular polarized light. The same thing, we consider two components, X and Y, but the trick is that for that light, we have shifted phase between these two components, and as a result, so these two amplitudes in X and Y will move or vibrate not in phase. As a result, on the detector, we will have circular polarized light. Depending on the phase shift, we will have right circular polarized light, or if it's, for example, shifted forward, so we will have a left circularly polarized light. And it's also perfectly described in terms of mathematics, in terms of Jones formalism, and this is a vector that we typically consider. Why? Actually, this vector is important. So the thing is, it relates clearly to the measurements. Basically, on my detector, I'm able to measure only X and Y components or intensity, so it means field in square. So thus, the first measurements, we do total intensity for coin cross. Then we do the difference between X and Y components. Then we rotate our detector for 45 degrees and able to measure the, this difference. And then we're able to see is there difference between left circle and right circular polarization. And these four measurements form so-called Stokes vector. And the Stokes vector is a quantitative measure of any state of polarization. So means by analyzing these four components, I'm able to describe any state of polarization. And typically, we use Poincare sphere that graphically represent the state of polarization. It's very simple, actually. Sounds might be crazy, but it's very simple. So the left circular polarized light, for example, considered on one of the pole. On the equator is the linear polarization. And between equator and poles, the left or right-handed elliptically polarized light. Means when these two components are not equal to each other. And then when we think, okay, when the light propagates to biological tissue, so there is a standard way to link these two components. So this is incident light, that's a light that's coming out from the medium, and the Mueller matrix, so-called, used to analyze quantitatively how the tissue influence the state of the light. And this is actually quite known for a number of years in terms of analysis of surface and thin films, and it's called polarimetry. And it's absolutely the same approach. And you see, so the number of books that was describing that technique. But again, the analysis of this tissue is based on the Mueller matrix component. And indeed, so when we use that technique, we're able to see for the tissue, so we're able to see and analyze that for elements of Mueller matrix images. And we're able to see the difference in terms of elements of Mueller matrix. And thus, there are at the moment number of results, and including results in our groups, and we're able to differentiate with a particular sample the healthy and unhealthy places with analyzing just the Mueller matrix. What is the problem actually here? So it works, but we have there is some problem. What is the problem actually? First of all, the question always, what is the optimal technical parameters for the system? Imagine just a simple schematical representation of how we can illuminate the tissue. For example, one of here. So, and what here? Imagine, so if we illuminate the tissue with one way, so the light coming to the tissue, so the detected light that comes through the tissue will have a bit different path length within the tissue. And that gives certainly uncertainty in our results, and we should know how to make interpretation of the results. And the interpretation of the results is very important, especially in the clinical stuff, because if we have some unknown phenomena, so we know what is that and how it happened really. So, and the other question was for us to test, are we able to use this polarization technology to test the tissues itself, ideally in vivo? But we started with these paraffin-embedded tissue blocks. So the problem is that some tissues, like human skin, for example, here have a very complex heterogeneous structure, and we should understand, okay, so what the outcome from different parts of the tissue we see in the polarized light. Okay, so thus we have a number of studies focusing on the peculiarities of propagation of polarized light and circular polarized light in the turbid tissue-like medium. And here's just a summary. So again, if you think about polarized light and it propagates through the empty medium, like a glass, so nothing happened. But if medium is absorbed some light, so you see the amplitude will be, becomes smaller. Okay, similarly happened for the circular polarized light. Amplitude is also changed. Okay, so, and if we look into some more complicated cases, oops. Okay, so Yeah, so you see, so these components that propagate through the medium will produce quite strange changes of the polarization. And it's very often happen for birefringent media, like a connective tissues. And we know we should understand, how the signature is formed, and if we know that, so we're able to detect, from that signature, what happened. The other well-known property of polarized light, imagine when we consider interaction of polarized light with boundary of the medium. So for different orientation of wave, so we will have slightly different reflection that is well-known from the Fresnel reflection, basic physics. So basically, we will see the difference in the amplitude of reflected light. But also, which is important, we will see the difference in the phase, means when the light incident on the different angle and reflected or refracted, then it's the phase shift between two components has happened, and we're not able to say clearly, so why, what we really see, the changes on the boundary, or is it because of birefringence? So this is just an example, well known. So when we have a wave, linear polarized light, coming to the medium, when it reflects, so one component will preserve the direction, another will be flipped, changed. So, and same happened with when we consider circular polarized light. So we have two components, those light that propagate through the medium, through the boundary, so will preserve helicity, and those light that reflected will change helicity. So it will becomes a light circular polarized light. So, and why it's important actually? So imagine, so if we send that light, it reflects with different helicity, propagating with a preserved helicity. And interesting that when we consider the scattering on the particles, same thing has happened for circular polarized light. If it's scattered forward, it's preserved helicity. When it's scattered back or side, so it's changed helicity. And thus, if we think how the light propagates through the medium, we always have a different scenario. So we imagine, so we illuminate the medium with a right circular polarized light. When it propagates through, part of the light will reflect and change helicity. Those light that propagates forward will preserve helicity. Those light that scattered at least one back scattered, so will change helicity again. And if it's changed back scattered twice, it will again preserve helicity. So first of all, that even and odd number of flips will be important here. But on the other hand, the message is that on the detector, we will always have two parts of the light. The one part of the light that preserved helicity, and another part of the light that changed helicity. And you see, so if these two components are equal, the sum of these two components will give us perfectly polarized, linear polarized light. That's a blue line here. If one of the component is prevail, so we will see elliptically polarized light, okay? And now, if we consider the non-cancer and cancer stage, imagine if we consider those cells at normal condition, and light is basically scattered on the nucleus. So this scattering regime is described by release scattering, means it's uniformly scattered anywhere. So as soon as the cancer has happened and DNA changed somehow, I don't know how they changed, but from the physical point of view, I know that the electrical constant will be changed slightly. And thus, that will affect the changes of refractive index of nucleus, and sometimes size as well. And as soon as it's happened, the scattering becomes me-scattering. And thus, here is it just shows you what is the difference. This slide shows the scattering for the normal tissue, when it's release scattering. And as soon as the size is changed, increased diameter, you see, so the forward scattering here is increased, means that most photon will propagate forward and preserve the helicity in our case, okay? So in reality, of course, it's more complicated because when we change the size of the nucleus, we have multiple flips, but it's actually can be managed and counted in terms of me-scattering, and we're able to understand how many flips have happened for different size of the particles. And also, this is the results of our computational studies. We demonstrated that when a number of photons that injected to the medium with the release scattering. So when they scattered and then averaged here, so the light will be distributed like and mostly becomes linear polarized. Whereas for concept case, it's more random variations, so we're not able to say that it's linear polarized. So that was also confirmed by results of Monte Carlo and the results of phantom measurements. And also we have studied a lot the localization of light within the tissue with the idea, that we're able to scan the particular area within and not, propagating down to the sample, because actually the samples from the biopsy are quite very thin. And this is just a schematic presentation of our results that produce circular polarized light. Light propagates, and we're able to see with this red color the area that most responsible for the signal for that particular source detector configuration. And as you can see, so by simply just by variate angles, we're able to control this area and produce quite shallow detection up to 500 microns in that case. So this is actually how the system is, prototype of the system looks like. It's quite compact. So this is the laser. Sometimes we use another laser, and the students, it's all done by the students actually. So, and we also suggested interesting idea. We believe that we use now the Poincare sphere as a graphical tool for quantitative measure of changes of polarization. And that, for example, was for different concentrations that shows clearly that polarization state is changed, and we're able to grade it in terms of concentration. So we also did it for some connective tissues and chicken skin and others, and also able to observe quite clearly the changes of polarization vector. And finally, so we checked it with the samples. And okay, so this red area was confirmed by pathologists that there is a cancer represented. So, and the green part is also confirmed by pathologists that they're like healthy, mostly the healthy tissue, so with no cancer cells. And it's and when we scan it with our technique, we're able to see the difference, recognizable difference between the Stokes parameter and Poincare sphere. And also, we demonstrated that this approach can be used to grade the aggressiveness of the cancer. So, for example, there's two different stages, and they clearly represented with good statistics. So it's published, certainly, sorry. And some results of the studies you can find in one of the recent issue from April in Journal of Biophotonics, where this story is clearly described. So, and in conclusions, what I should say that, first of all, we claim that circularly and elliptically polarized light scattered within the tissue is highly sensitive for presence of cancer cells and their aggressiveness. And the changes of anisotropy of scattering particles can be clearly observed by tracking Stokes vector of circular polarized light on Poincare sphere. And theoretical studies show that helicity of backscattered circular polarized light depends on anisotropy and on the size of scattering particles. And now we're working on the development of Poincare sphere as a navigation tool. And just a few words about that. So that's our further development. So, and also what we see is in the future. Imagine, so when we discussed here, we considered the light that have a simple, we called circular elliptical polarization. But there is a special light that have also phase changes across the beam. So, for example, if we consider that it's called Gaussian beams or vector beams, and here, if I consider how it propagates, the orientation of polarization there will be changed across the beam, and like that, you see. So when light propagates, so we have a clear angular momentum there, and we demonstrated that this light is more effective to observe the depolarization and phase changes when it propagates through the medium. So that's at the moment the results of theoretical studies. This is just another configuration of Gaussian beam, but it's clearly seen that patterns could be analyzed quantitatively. And also, we think that point sphere could be used as like in a color code and able to link each position with a position on the surface of the sample, and that gives us quantitatively the parameters of polarization light that could be linked with tissue. So thus, in future, we believe that point sphere could be used as a graphical tool for tissue characterization. Imagine if we see, let's say, here, the red part and saying, okay, so in Brazil, so there are no cancer. We say, okay, on the North Pole here, so there are different colors, so there is a cancer. So that's just one of the, like a GPS navigator. Okay, so this is just some pictures from our activities. And we worked on the polarized light, but we also link this with the, we link this with the different other technologies like imaging and laser speckle technologies and so on. So, and also with the development of these calculations of light propagation through the medium, so we developed so-called online Monte Carlo tool that's available online, and you're able here to use free of charge the main computational tasks that's required for in the biophotonics, basically. And we have quite sufficient number of users, so around the world, that daily count something. So, and you see, so in Brazil, so it's interesting to see the distribution of those people who worked with biophotonics and especially Monte Carlo. But I'm particular always, like that here, the little island. So, and I would like to visit it sometime, to see who is actually doing Monte Carlo there. Okay, so this is the students all from our group. And this is our pathologist, Mike Eccles, who is working in Otago Medical School, and he gave us this, all the samples. And we're also happy that in the last year, so we have the two great physicists from United States, Stephen Jacques and Bob Alfana, who also involved in, with us in these studies. And we think that on the next conference, we will provide more sophisticated results. Thank you for your attention. If you have questions, I will be pleased to answer.
Okay, so the presentation can have a few questions. Well, let me ask one question. When you explore the Poincaré mapping stuff, can you do the dynamics? I mean, how the tissue is changing? So if I do many images and keep accumulating?
Yes, and there was one picture that shows the dynamics, how the number of photons represent different one, and then when we average them, so they will show you a particular point. Yes, that's possible.
So if you don't have any more questions, you always can ask Igor to talk with you during the interval and everything.